We'd like to welcome the former FDA Commissioner, Dr. Scott Gottlieb, who will explore investing in better health and the power of preparedness with CNN's award-winning Chief Medical Correspondent and Neurosurgeon, Dr. Sanjay Gupta, and Dr. Albert Bourla, the Chairman and CEO of Pfizer.
Hello, I'm Dr. Scott Gottlieb here at the International Vatican Conference. The global response to COVID-19 has been accelerated by extraordinary collaboration in data sharing, in research, and in epidemiological surveillance. Joining us here today to talk about how we can improve future response and build on a lot of the collaborations that have been forged in response to COVID-19, and what we can build on learning about preparedness for the future, on vaccine development, on distribution, on issues of surveillance, are Dr. Sanjay Gupta from CNN and Dr. Albert Bourla from Pfizer, where I have the pleasure of serving on the board of directors. It's great to be here with both of you. Albert, I wanted to start with you, if I can. In developing the vaccine that Pfizer pioneered this year, we saw new kinds of collaborations, not just in how vaccines were developed, but in how other therapeutics were advanced as well. Some of them very novel. Can you talk about some of those collaborations that you saw being forged this year, and how big of a role did it play in speeding up discovery and development of the vaccine and other therapeutics that I know Pfizer's working on?
Of course, Scott. And first, let me say what a great honor it is for me to be in the same panel with Sanjay and you. I think that in times of crisis, they say that characters are tested. I think also the cultures of corporations are tested. And I think that was a great test for the entire biotechnology, pharmaceutical, and regulators ecosystem. And I think all of us, thank God, we passed with flying colors. There were a lot of collaborations that had to be in place, and many of them were typical, and a lot of them were atypical in the way that we work. I would say that one of the most atypical collaboration is the one that we had ourselves with BioNTech. Not because we partner and we join forces to develop a vaccine. That's very common. We do that with a lot of therapeutics and vaccines. And this is how science is progressing, by joining forces. But we did that without having a contract. And I don't know if many people know that when we decided to join, I spoke with the CEO of BioNTech, my dear friend now, Ugur Sahin, and I told him, Ugur, understand that we are running against time. We will not have time for contracts before months. And he said, I understand that. Can we shake the hands through the Zoom camera? And this is how things went. We started, they shared their intellectual property. We shared our intellectual property with them. We placed billions into development. And the only thing that we were worried was, what is the next decision to make sure that the vaccine will be effective and safe? The vaccine had positive results in November 20. It was registered in December of 20. You know when we signed the commercial agreement? In January of 2021. Agreement that will gather billions of dollars were just put on hold just to make sure that we are all focusing on making the vaccine happen. I think these are the examples that I'm sure exist elsewhere in the industry that make all of us so proud.
Thanks for that, Albert. Sanjay, I want to put the same question to you, sort of in a broader sense. You sit across a lot of different venues. You see what's going on in the industry. What lessons do you think researchers have learned from the collaboration that's gone on in the context of COVID? And do you think we're going to be able to continue to collaborate with government among private entities going forward? Have new models been forged?
Yeah, well, first of all, let me just say thank you as well for having me. And Scott, this is a real treat answering the questions from you. I've always enjoyed asking you the questions, so I appreciate this. And Albert, what an honor. Some of what you just described there is really fascinating. I've been covering this as a reporter for well over a year now, but you learn more about some of the background of this every day, it seems. Yeah, what I would say, Scott, to your question is that I think the public really had no idea, most of the public at least, how these collaborations work or how they come together at all. Whether it's Pfizer and BioNTech or the NIH and Moderna, whatever it may be, people got a real sort of granular look at a lot of this. I mean, there's a lot that they still don't know, but I think it's very interesting for people. And as a result, the end product was something that I think is unquestionably being celebrated. The idea that these vaccines could be developed at this sort of speed and the pace of medical innovation has been forever accelerated, I think is something that people really talk about, they celebrate, they're enthusiastic about, and now I think they will come to expect, which I guess is part of the answer to your question. I think it will change because it's now been laid bare in a good way for people to see. So that I think is some form of that is probably here to stay. I think there's all these lessons going forward that people, everybody will sort of offer up in terms of, what this means going forward, but I think it's here to stay.
Albert, can you talk a little bit about the mRNA platform, developing it, what you think it means for the future? Is it something that's going to be applied to other therapeutics, other vaccines? What does the future of R&D look like with mRNA?
I believe so. I think it's a technology that is maturing for the last 20 years. Ourselves are engaged with this one in the last 4 years. And we had to advance this technology, I think, by 10 years within one. That's the speed with which things were progressed. So right now we have reached a level that we understand so much about the nuances of this, that the field of applying it to therapeutics or other vaccines is wide open. I believe that we will see tremendous activity from multiple biotechnology companies right now, working on different type of vaccines and working on different type of therapeutics, like therapies for cancer or therapies for genetically driven diseases. And Pfizer, of course, is one of them. We are very, very eager, and we have jumped already in multiple different applications of this technology.
Can I just add something as well? I had a chance to talk to Albert's dear friend, Ugur Sahin and Özlem Türeci, his wife, who are the couple behind, BioNTech, and a fascinating couple. Everyone should just read about them. Their honeymoon was going back to the lab right after they got married. That's the kind of couple they are. But so much of the work that they've been doing on mRNA, as Albert mentioned, was around cancer prior to this. There was some of their efforts towards vaccines, but so much of what Ugur was describing to me was around cancer. They hadn't quite gotten there yet. And then there was obviously a sudden pivot in January of last year. But the way that platform works, could work, I should say, I just found really intriguing. I mean, this is not my area of expertise, but when we talked for 2 hours about how these mRNA vaccines, or how these mRNA platforms, I should say, could induce an immune response for certain types of heterogeneous cancers. I mean, if we have the same sort of, outcomes that we had with these vaccines, I just found that very, very inspiring.
Sanjay, you sit above the industry, you've followed this for a long time. Do you think that the work we've done in the context of COVID is going to help validate the platforms or help accelerate their potential use in other therapeutic areas?
I really do. I mean, I think that what has struck me the most here is, having been a medical journalist for 20 years, there was often a didactic nature to things. I think, based on 10 years of data, here's what, we're telling you. Here, what we saw was the world sort of learned a lot about this together. I didn't know anything about mRNA platforms prior to starting to report on it. I had to just learn a lot. Everyone did. And so I think that degree of collective learning, I think, is going to make people very interested in this, feel a little bit of ownership over this, brag that they can talk about this. I hear non-scientists describing this to their friends all the time. I love that. You do have a quarter of the country roughly that's hesitant for the vaccines, but I think the larger picture is they're hesitant about something that seems too new, or, okay, you told me it was going to take a long time. It took a short time. So that's got to be a concern, all those sorts of things. And I think that's got, everyone says this, but that's got to be addressed. Maybe the way to address it is really letting people in on the science. Here's it's just a little strand of genetic material. Here's what it does. It's a little Snapchat. It disappears in your body. Whatever it might be, to continue to hammer those points home will make a difference. It won't solve the problem, but I think it'll make a difference.
Albert, I wanted to come back to just the continued development work, and we're obviously seeing variants around the world that are raising concerns that we could have different phases of the pandemic if new variants become more common. How quickly do you think your labs or labs generally can be able to develop boosters to combat these variants? The one that we're seeing in South Africa, the one we're now seeing in India and elsewhere.
I think very fast, because we have right now already ongoing trials. As far as our vaccine, we have now data that the current vaccine works against the South African variant. Our study in South Africa had 100% efficacy. There was smaller sample, but 100% is 100%. Our real world data on the UK variant, which is the most dominant variant right now. And I hate that I call them with the name of the cities or the countries, but this is how most people know them. It was the most prevalent variant in Israel, and over the 85% of the cases were from the UK mutation, and we had 97% efficacy there. So everything we know it is that the current variants are covered by our vaccine, but nevertheless, we are trying the efficacy of a booster shot, and we are trying the efficacy of a booster shot of the same vaccine against the wild type and all the variants, and also the efficacy of a new vaccine, which is tailor-made for the South African variant against again the variants and the wild type. And this data will come in 1 month or two, so it's really imminent. And right now, I think what is the important it is that we know that the possibility that the variant that will escape, that will evade the protection of the vaccine is real. So that can come anytime. So as a result, what is important it is that you have a system, that you survey all variants, and that you have readiness, that you will be able to develop, if needed, a booster vaccine in very short period of time. And right now, I'm happy to say that we have our process is geared to do that in less than 100 days. Right now, we are already 90-something days, that we should be able, from the day we make the decision that this is a variant that we need to do something about it, within 90-something days, to be able to have a product ready.
I should just comment, Albert, you've been outstanding at keeping the board of directors, including myself, informed through this crisis on Pfizer's ongoing work. So I am putting you in the unfair position of asking you questions to elicit information I already know, but I want the audience to know. So I thank you for playing along.
Can I ask something that I don't know, because you both know all this stuff so well, but the boosters that you're talking about, especially the ones that are variant specific, do they have to be reauthorized, or how would that be handled from a regulatory standpoint?
Well, yes, they have to be reauthorized. I think that should be a very different process than a whole new registration, but we have a ton of expertise because, or there is already established regulatory pathways because of flu. In flu, you have a registered vaccine, and then every year you need to change the strain. And there is a process what you need to do so that regulators around the world will approve it or not. But you have the best regulatory expert next to you. I think that comes to Scott.
Yeah, in the context of flu, Sanjay, you're looking at the immunogenicity induced by the vaccine against sera. You're not even looking serum. You're not even looking at clinical data. So you're looking at how the vaccines are able to produce antibodies that target the specific epitope that you're trying to enlist a response against. And so in the context of coronavirus, I think what you're going to see emerge, and it's going to evolve over time, but I think what you're going to see emerge is the ability to authorize new vaccines on the basis of immunogenicity data, the ability to demonstrate that you can develop antibodies that bind to the new epitope, so the particular spike protein that's in 1351 or B117, without looking at outcome studies. The FDA will rely on the original finding of safety and effectiveness from the original studies on the overall vaccine construct, and just be looking at the ability to generate specific antibodies to specific epitopes with some of these new variant vaccines. I think that that's the construct that they'll come up with. So they'll probably still require some clinical data to look at antibody response in people, but they won't be looking for long-term outcomes in those studies.
So go fast.
And if I can add here, one of the reasons why, for example, I was convinced let's go with the mRNA technology was because of how flexible this technology is, to be able to adapt to changes and to adapt your vaccine to changes of the variant. One aspect is manufacturing, for example. Right now, we have a process that we will lose not a single dose. So the machine or the whole process will produce the current one, and then very quickly, without any delays to adjust, we'll start producing the new one, and we will just replace the current doses with the new ones. This is not the case with all the technologies that you need to start and go through the whole new process. And so it's very, very dynamic, and it is really tailor-made for something like that.
Yeah, and Sanjay, you'll remember in the swine flu, in 2009, one of the rate limiting steps, one of the reasons why the vaccines were delayed, the flu vaccines that we developed that season, was because there was difficulty getting that new strain to grow in cell cultures, and the yield ended up being much lower than what the companies were anticipating. So, you know, the rate limiting step in the past has been the ability to culture the virus and get it to grow in cell culture in the yields that you need to provide a sufficient amount of vaccine. Here, it's, you know, Albert's described it to me as sort of plug and play. If you come up with a different mRNA construct, you can basically put it into the existing manufacturing processes and have a reasonable degree of assuredness that you're going to get the same yields and the same output. Did I say that right, Albert?
You said very well. Actually, I asked our head of manufacturing, so how much are we going to make? At least 2.5 billion doses this year. If we have to switch mid-year to the new vaccine, how many doses are we going to make? 2.5 billion doses. Incredible. It's going to be just plug and play.
Sanjay, I want to get you back into the mode of being someone who answers questions and doesn't ask them. So I'm going to put a question.
Can't help myself.
yeah, you can't.
You can't help yourself, Sanjay.
You've covered a lot of emergencies, a lot of epidemics in the past, disasters over 2 decades. You've been doing this. How does this sort of fit into that picture? I mean, obviously, it's got to rank up at the top in terms of severity. How do you compare it to what you've seen in the past?
This was far and away the most intense from a coverage standpoint that I think I've ever had, and I think there's a few reasons for that. One is that, you know, obviously, really since, you know, January, maybe even late December, because we were sort of monitoring what was happening in China in December of 19. We've been at this. I mean, all of us have, obviously. So, but you know, it's just been just relentless, really, in terms of the coverage. And I think what journalists often do is I wake up very early, you know, before 5 o'clock in the morning every day, so I can talk to some folks on the other side of the planet who may be ending their day, and, you know, really just trying to learn as much as I can about something that was so novel, despite the fact that I've covered outbreaks and epidemics before, and even a pandemic like H1N1. This just was different. I think on a more personal note, nature of a pandemic is, I guess, no one is really insulated from this, you know, and so I'm dealing with I have 3 girls, age 12 to 15. By the way, I do want to know when they can get vaccinated. I'll ask that later. But 3 girls, and, you know, we decided to keep them home from school for a while, you know, worried about what was happening and just navigating all the realities of life through this. I think when you cover conflicts or natural disasters like Haiti or Pakistan or Japan, it can be intense, but you know you're coming home at some point, and I'm going to sleep in my bed and it's going to be a different world. Here, we were all just very immersed in it. I liken it more, I think, Scott, you'll appreciate, to chief year of residency in some way. My chief year of residency, all I wanted to talk about and learn about was the brain and the spinal cord. That's it. I really didn't have a lot of time or interest in being social or talking about other things. It's kind of been that this year as well. My conversations are about this virus and all the work that you guys are doing. I've not been very social, I think, probably like you guys as well. So that's been very different personally and professionally.
Well, thanks for that. Thanks for your time today, Albert. Thanks, Sanjay. It's been a pleasure to join with both of you today. Thanks a lot.
An honor. Thank you.
An honor. Thank you.